Welcome. This will likely be the most impactful YouTube video you'll watch this year on aquaculture and the aquarium hobby. Today we're going to talk about thyroid disease in fish, as well as the mechanisms by which thyroid disease and nitrate impact immune function in fish. This is applicable to both the aquaculture and aquarium hobby as a whole. All of this information is cited from peer-reviewed sources, and the citation will appear on the screen as I share the information. You can also look at the description of this video where I will have all of the citations that I used to create this video. I highly recommend reading them if you're interested in the topics I talked about during this video. During the course of this video, I will talk about a few topics, including why thyroid disease in fish is important to know about, the way the active thyroid hormone T3, is formed, nitrate's impact on thyroid hormone 3 and 4's impact on vitamin A metabolism, the impact vitamin A deficiency has on immune function, iodine deficiency and the results of it in fish, why you should never, and I mean never, get an ozone generator for your marine aquarium, and finally, how thyroid disease presents in fish. In short, we're going to cover a lot in this video. So why is it important to know about thyroid disease in fish? Not only is it likely to have a major impact on aquaculture and the aquarium hobby as a whole, it is a vital regulator for many things within fish. They're involved in the metabolism of fat, proteins, and carbs as an energy source. They increase the breakdown of fat in the liver, and they have a major impact on growth, reproduction, and coloration. Those last three topics will be the topics of other videos related to thyroid disease in fish. And finally, thyroid hormones play an integral role in immune function, which is the big reason why it's so essential to know about and understand. Now let's go over how the active and essential T3 thyroid hormone is created in fish. It starts with the hypothalamus, releasing thyrotropin releasing hormone, or TRH. TRH then acts on the pars distalis, which is part of the anterior pituitary gland. This triggers the pars distalis to release thyroid stimulating hormone, or TSH. TSH stimulates the thyroid to produce T4 thyroid hormone. The way it creates the T4 hormone, it stimulates the thyroid follicle to uptake inorganic iodine from the bloodstream of the fish. It then combines the iodine with the protein thyroglobin to produce the thyroid hormone T4, and then after it's created, T4 thyroid hormone is stored in the lumen of the follicle. After that's done, T4 is converted to the biologically active T3 thyroid hormone. This occurs in the liver and possibly other organs, and those are the immediate steps in the production of T3 thyroid hormone. Now let's talk about nitrate toxicity in relation to T3 and T4. The main impact that nitrate has on T3 and T4 is that nitrate inhibits iodine uptake at the level of the thyroid follicle. And based on what was previously just said, you know that iodine is involved in the creation of T4 and T3 thyroid hormone. It is an essential limiting nutrient for it. That means that the production of T4 thyroid hormone is inhibited with reducing iodine levels, and without T4, T3 cannot be produced through conversion, and the active and most important thyroid hormone, T3, is not formed. On top of that, it is speculated that nitrate may inhibit the conversion of T4 to T3 via an enzyme, which is an important mechanism for thyroid hormone regulation in fish. As a result, T4 is then lacking, and thyroid stimulating hormone goes crazy, inducing goiter and a hypothyroid state. Lastly, while it hasn't been observed in the majority of fish species, sharks have been known to develop hyperplastic goiter, which is the common cause of hypothyroidism in fish. It is produced as a result of nitrate exposure 70 part per million for 28 days. After that 28-day period with that nitrate level, white spotted bamboo sharks were shown to have hyperplastic goiter and be put in a hypothyroid state. Now let's talk about the impact that low T3 and T4 thyroid hormone has on vitamin A metabolism. The study above fed cabbage and thyrouracil to grass carp. The reason that they fed grass carp thyrouracil is because it's a potent inhibitor of T4 and T3 thyroid hormone. It inhibits these hormones by stopping the absorption and metabolism of iodine, which results in an iodine deficiency in grass carp shortly after the experiment begins. After the experiment was over, they measured the concentration of T4 and T3 thyroid hormone from the blood and measured 3 vitamin A sources from the liver of the grass carp. The findings of this study clearly showed that the metabolism of vitamin A sources are significantly connected to low T3 and T4 thyroid hormone levels. A significant decrease in T3 and T4 thyroid hormone was seen in the cabbage and thoracil group. Then the same fish were examined to see the vitamin A concentration in their liver. It was seen that both the cabbage and thoracil group, in those two groups, the vitamin A concentration was decreased by a considerable amount, and it corresponded to the decrease in T3 and T4 thyroid hormone, giving strong evidence that impeded thyroid hormone production in fish has a negative impact on vitamin A metabolism. Now let's talk about the impact that vitamin A deficiency has on the immune function in fish. Vitamin A is a vital vitamin for immune function. Let's go over some of the reasons it's so vital now. In an experiment, 5 groups of fish were fed 5 different diets with varying concentrations of vitamin A. The groups included a 0 vitamin A group, a 2500 vitamin A group, a 5000 vitamin A group, a 10,000 vitamin A group, and a 20,000 vitamin A group. This is all in IUs. They were fed these diets exclusively for a 10-week period. At the end of the 10-week feeding trial, the Nile tilapia fed the unsupplemented diet had significantly reduced white blood cell counts compared to the second lowest group, 2500. This shows that with a decrease in dietary vitamin A, there is also a noticeable decline in immune function via white blood cell count. Another metric we can look at is serum protein concentration. Serum protein is very integral to many functions within fish, including nutrient transport within fish, and it's also very important for maintaining immune function and overall health in your fish. In the unsupplemented diet, the serum protein concentrations were significantly lower than those who were fed a vitamin A diet of 20,000 IUs, and the value for this metric was moderate for the 2500 and 10,000 IU group. A third metric we can look at is lysosome activity. It appears that lysosome activity was the highest in the 5,000 IU group and lowest in the heavily supplemented 20,000 IU group. This implies that not only does vitamin A deficiency have an impact on immune system function via white blood cell count, but an extreme excess of vitamin A can also have a negative impact on lysosome activity. Lysosomes are an important part of the immune system and very capable when combating bacterial infections in fish. They both kill and defend against bacterial infections, and low lysosome activity will put fish at a higher risk of bacterial infections. Overall, I think that this section shows that vitamin A is highly interconnected with immune function in fish, and that a deficiency or extreme excess of vitamin A would have negative impacts on your fish's immune function. Now let's talk about iodine deficiency in fish and the effects that it has on them. But first, let's point out the biggest reason why iodine is so vital for fish. Without iodine in the bloodstream, T4 cannot be produced, and iodine, as a result, is an essential limiting nutrient for thyroid hormone production. And then when a fish has insufficient iodine, thyroid hormones are unable to synthesize T4 correctly and become overstimulated by thyroid stimulating hormone. This is because T4 has a negative feedback inhibition loop, which just means when T4 rises, it tells thyroid stimulating hormone to stop. And when T4 is too low, thyroid stimulating hormone goes nuts. When thyroid stimulating hormone continues to be produced within thyroid follicular cells, the thyroid follicles then become enlarged from hyperplasia. Now let's talk about why you should never, and I mean never, add an ozone generator to your saltwater aquarium. In saltwater aquariums, iodine exists in 2 forms, iodide and iodate. The form iodide is biologically active for uptake within the fish's gills, as was their gastrointestinal tract. On the other hand, iodate is simply not biologically active and able to be accumulated through the gills and through their diet. In a saltwater aquarium, an ozone generator is used to reduce the bacterial load in your aquarium, in particular to reduce harmful pathogens that may make your fish sick. But how effective this is often debated by hobbyists. Ozone generators, however, drive biologically active iodine and turn it into iodate in considerable amounts, which then reduces the amount of bioavailable iodide in your saltwater system. When ozone generators are used at the recommended amounts in saltwater aquariums, they drive a significant amount of iodide into iodate, and this drastically increases the risk of iodine deficiency in fish. In fact, evidence of goiter has been shown in brown-banded bamboo sharks as well as white-spotted bamboo sharks within 60 days of adding an ozone generator. For all fish, adding an ozone generator into an aquarium is a risk, but especially for those species of fish that prey on seagrasses and kelp, such as tangs and other species. These species will need more iodide in their water compared to other ones. And if they cannot get their iodide through their diet or their water column, they will develop hypothyroidism and become sick as a result. And finally, let's go over some of the clinical signs of hypothyroidism in fish. Hypothyroidism may occur secondarily to an impaired ability to move and articulate the jaw, and can include what is referred to as gapping, which is when a fish's mouth is stuck open and cannot be closed, and as a result, it hangs open chronically. In less severe cases of this, the jaw may just move erratically. This can also cause difficulty in breathing, which is attributed to the thickness of the brachial arches of the enlarged thyroid gland. And finally, the impact that T3 and T4 thyroid hormone has on the pigment in fish. This will be covered more in depth in another video. Black pigment cells are called melanophores, and red pigment cells are called erythrophores. Both of these pigment cells have been shown to increase with a decrease in T3 and T4 production. On the opposite end of the spectrum, blue cells called iridophores, far red cells called erythroiridophores, silver-white pigment cells which are again a form of iridophore, and yellow pigment cells called xanthophores. These have all been shown to decrease in abundance with a decrease of thyroid hormone T3 and T4. And thanks to this impact that it has on pigment, we can partially gauge thyroid hormone health by the color of your fish. All of the symptoms I just mentioned are signs that your fish could have goiter, aka hypothyroidism. And that has been my video detailing what we know about thyroid disease in fish, which is likely a widespread problem in the aquarium hobby, and one which most people will always be unaware of, but hopefully this can change that. And as I said earlier, all of the sources I used to create this video are cited in the description.